Hello, AP class. How are you? I hope you are all doing well. Today we are finishing 4.1 on the actual population biology or biology population, however you want to say it. But today we're going to kind of finish up this lecture. Do know that there's some key understanding points in this lecture, so which hint, hint, hint will maybe hop up on a quiz or exam. So it's important that you understand them and know the importance of them, but also the limitations of them. And we're really going to start off today about, it's all about, obviously this lecture is about population and keep, understanding what certain populations of given animals are. Now you can think about, why, before we begin, why is that important? Why is understanding how much of a given species there is out there important? Think about that, right? Not just for humans, but what about endangered species or species that are, this close to extinction or provide, such as honeybees? Why is, accurately trying to predict how many honeybees there are left important to the world around us, right? And hopefully you can kind of build that up in your, your mindset of why we need to understand populations of just not humans, but other animals, right? To see how the environment is doing and to see how maybe, healthy our world is. So how do we do that? As scientists, how do we estimate the population size? Well, one of the biggest ones we'll talk about in AP Environmental Science is mark, release, and recapture. One second, I just want to change that real quick. All right, sorry about that. I had to turn off some music that was in the background. But the main way we'll kind of talk about in this course is the mark, release, and recapture. It's important to understand, the scientific methods of how scientists do things, and this is one of them, which we will kind of explain. And we'll see the good for this, as well as you should understand the bad or why we don't necessarily use it all the time. But mark, release, recapture, or MRR, is the first step is to capture and mark samples of individuals. So we literally go out and we give a certain area of land and we say, okay, let's see how many, using these photos, for example, how many field mice are within this acre or rye of land, right? And then that way we can then estimate if we have, this many in this amount of rye land. Well, in 100 rye there might be this many, or in 100 acres there would probably be this many. Right? Now, of course, you can probably see limitations to this and how it's not 100% accurate and often is not. Do know my goal for you is to do a lab based on this, which will come after the exam, but do know that will be coming up. That will kind of play around with this idea and the actual name for it. But then, so we capture them, we mark them. It's important to mark them not where it will harm the animal, right? I would even say some of these methods that you see in the photos, probably not the best, but scientists are trying to do their best, right? But you want to make sure that it does not in any way hinder or harm the animal, right? That's important. You don't want to hurt your test subjects who you're trying to save. It just doesn't make sense. So animals are immediately released as close as possible to the collection site. Why would that be important, do you think? Right? Well, you want them to continue on life as normal as it was. If you remove them somewhere else, well, then it skews up your data. Give animals time to recover, and then randomly mix with population, then go out and you collect a second sample of organisms. This is called, right here, this is another example of, tagging an animal. You see this big box on them. This way you can kind of locate it, which now looks quite primitive to some of the methods we'll talk about that some scientists are utilizing now today. But what this is called is the Lincoln-Peterson index. This is how we do it, and you definitely need to know this formula to see what a actual the number of a given population size is. So size of population can then be estimated from the number of marked individuals recaptured on the second day. So we have the Lincoln-Peterson index. Now look at this formula quite closely. N, you're trying to find capital N, right? That is the total population. You're trying to find what the total population of this given area is. N1 is the number of animals captured on the first day. So simply, you capture, you go out, you set aside a square acre of land, you go out to catch turtles, right? You find, well, let's say birds, you capture 20 birds, so then your number would be 20 there. You mark all those 20 birds and then you let them go, right? That is kind of what we're doing here to make sure they're safe and sound. Then you allow some time to pass so you can let those birds resettle and get used to their environment. Then you go back in and do a second gathering. Well, some of those birds you capture on the second gathering will, and some will not, have that tag. That is the number, and you put the total number of birds you caught on the second day. But M represents the numbers of marked animals that you recaptured. So let's say you go out on that second day and you find, 30 birds this time, but 15 of them have the tags, 15 of them do not. Well, M would be 15 for the amount that does have the tag, and then you run it through this formula. So you take the first day times the number for the second day, and then divide it by the number of re-tagged animals. Ladies and gentlemen, please know that this is one of the scientific instruments that you have to know for AP Environmental Science. So learn it, know it well. Like I said, we will be doing a lab on this to kind of further strengthen your understanding of it, but it's imperative that you kind of get it, right? But kind of a word sample here. But suppose you want to know how many box turtles are in Lithia Park in Ashland, in Nebraska, actually my home state. On the first day, you hunt through the woods and capture 24 turtles. So N1 would be 24. You put 24 here. You place a spot of paint on each turtle shell and release all turtles back where you found them. A week later, you return and catch 60 turtles. So 24 is N1, 60 is now N2 because that's how many you caught the second day. So really you can multiply those, right? Of these, 15 are marked. So you have 15 marked, so that would be in M. What would be your estimate for the box turtle population of that given area? Go ahead and do the math for me. I'm going to do the math as well. And yes, you can use a calculator. What would your answer be? Hopefully you got 96 just like I did. And yes, I wrote it out here on my paper. But 96 would be the relative population of that given group of animals, box-shell turtles in a given area that you went out and hunted. So that's kind of how we can estimate the population size. Now, why do we use the Lincoln-Peterson index? Why don't we just go out there for days on days and days? I think I kind of answered my question, and just find every single turtle. Well, of course, that would take too much time. It would take too much work. You might not find all of them, right? You wouldn't get an accurate number anyway if you're searching for them. So this is just kind of a way to estimate the amount of turtles in a given area. Let's try another one. So a biologist nets 45 largemouth bass from a farm pond, tags their fins, and releases them unharmed. A week later, she then nets 58 bass from the pond, including 26 with tags. Based on the Lincoln Index, estimate the number of bass in the pond. So go ahead and do that for me. All right. So roughly you get 100 point something, 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 right? But obviously you just round that to the nearest number. So roughly you'd have 100 largemouth bass in the given area or that pond that you're searching for them. So hopefully that kind of helps you out with understanding Lincoln-Peterson index. But there are some, obviously we kind of talked about there are some negative aspects about it, or it's not 100% correct, right? So the Lincoln Index assumptions that you should know, right, or things that it doesn't take into account are individuals with marks have the same probability of survival, right? What if you have birds? What if you caught your birds and you put a big old green stamp on them? Well, that might make them a better target for their predators, right? So you have to make sure whatever you mark them with doesn't hinder them, right? Because that could then skew your numbers because then you have a lot loss of life, right? Or it might just be easier for you to re-find them again in the wild on your second route. 2, births and deaths do not occur in significant numbers between release and capture, right? Let's say you took that week to let those animals settle back in, but then there's a forest fire. Obviously, that's going to have some sort of hindrance on your data. Immigration, emigration do not occur, right? You're not taking account that, maybe it's a very migratory species. Those might be a bit harder because if you have a lot of coming and going, you don't have precise numbers. Marked individuals mix randomly with population, right? You want them, hopefully, if you let, all of your specimens out the first time, hopefully they actually go out and mix again, right? They don't just stay in the area you just left them, because then that would skew your data. Marked animals are not easier to capture a second time, right? That kind of goes back to if you could see them or if you hurt them, right? Make sure they're not easier to capture, because then your data would be skewed. Marks do not come off organisms, right? Then you wouldn't know which ones are tagged, obviously. Recapture rates are high enough to support accurate estimates, right? Let's say you go out and you get 60, snails the first capture, then you go out a second time and you catch none. Well, obviously your data is going to be quite skewed because you don't quite know what's going on there, right? So it's important to understand these things and know the limitations of the Lincoln-Peterson index. We will be doing a lab on it, so be prepared for that. That should come up right after your exam before school is out. Okay, so why is this important? Why is it important to know the population of animals? Well, we need to be able to protect them, right? And we need to know how many we have left. So there's a biology called conservation biology, which is the critical question in conservation biology: is the minimum population size of a species required for long-term viability? So I want to throw this idea at you first before we kind of go on to this slide or the next. Elon Musk has said that he wants to send humans out to space, right? He wants us to travel the world. His estimate or his goal was to get 10,000 people on Mars to have a viable colony. Why 10,000? Think about that. Why would we have 10,000 people? And his reasoning was that, if something happened to Earth, humans would be able to go on. But why 10,000? Well, that is the minimum population size for humans to go on. In regards to science, as long as we have about 10,000 individuals, the human species overall can survive healthily, right? If you have less than that, that means the human species is not going to have enough genetic differences to make it viable population size. So think about this now when we're talking about animals, right? Animals are losing their land extremely at a very high rate, right? We're losing forests at an extremely high rate. But also think about like the white rhino or different species that may only have like 3 specimens left. There's only 3 animals left of the whole entire species. Is that a viable population? Absolutely not. That population, that if we are able to save that species and they grow from those 3 individuals, they are not going to be the exact same or a whole look as the species was in the past. Why? You're going to get the genes from 3 species or 3 animals over and over and over again. It's going to be very inbred, right? It's going to have very genetic similarities. Animals like this tend to have much harder times in the environment. So not only are they the 3 last, but their offspring are going to find it much more difficult to survive in the world around them due to genetics, because they will not have a genetic ability to withstand certain requirements, right? Like some, putting this in human terms, some humans have genetics that make them less susceptible to cancer. Many or other people have genes that make them very susceptible to cancer. Well, you might say, well, those 2 people should probably get together and make a baby so that their baby can have those good genes, right? So that's kind of what we're getting at here. And then when we're kind of talking about it, special consideration, think of island biogeography, right? I already talked to you about there is no more really huge swaths of wild land anymore. Really what you have is these island little forests that now exist. Think about Thailand. You can't go, 10 kilometers without seeing some sort of developed land. You might see a bunch of forest in between, but then it's developed land. Think about tigers. So that tiger is stuck in that little swath of land where before it could move on and meet new, new relationships, right? It could form new relationships with other tigers, but now it's stuck in that one area. So if you may have an area that just has 2 or 3 tigers, it's the same thing. You're going to have a very species that's not very strong or capable of defending itself in the future, right? Because we have this habitat fragmentation going on in the world. So look at this. On the left side, we have, we're a bigger swath of land where you can have many different types of animals that can coexist in one area. Well, if you start breaking that up, well, maybe, the tigers get stuck in this one, the elephants get stuck in this one, and, maybe different types of animals need each other to survive, but now they don't have each other anymore. So you start to have this fragmentation, which is bad for the environment or for the species. In a large population, genetic diversity tends to be preserved, right? So if you have like 10,000 humans or more, genetic diversity tends to stay stable, right? Or you have enough genetic differences to keep it stable. But less than that, then you start having problems. A loss of gain of a few individuals has little effect on the total gene pool, right? But however, in small populations, small events can have large effects on the gene pool, right? Going back to this photo. Let's say we have, a forest fire that burns up this 1/8 of this forest, right? Well, some species are obviously going to be affected. But let's say you then break up this, which might be about half this one. Well, a lot more species are going to be affected. So there's a thing called, what we have is genetic drift, change in gene frequency due to a random event, right? A random event occurs, it might have effect on the gene frequency, or it might kill off certain genetics, right? If people with blonde hair live in a certain, if they were relegated to a certain building, dear God, let's hope humans never get to that point, right? But let's say you start dividing up different genetic people in different buildings, and then one of the buildings starts on fire. Well, then you're going to lose a whole genetic gene pool, right? And then we have also the founder effect, right? Think about a brand new island and the seeds come over from a brand new island. Obviously, those seeds are not going to be a genetic, if let's say it's a beautiful flower, it's not going to have the genetic diversity of that whole species of flower. It might just be from an individual. So all those new flowers on that island are going to now have just the genes of that given organism that sent the seeds, right? And that's what we call the demographic bottleneck. Just a few members of a species survive a catastrophic event such as natural disaster, and this reduces biodiversity and increases inbreeding, increase in recessive alleles. Mr. Bryan would beat me up for saying it like that. But let's say this is a great example, and I actually have a video on this to explain the whole point. So I do make sure that you do watch that video. I will attach next. But let's say this is what the regular population looks like, right? Like a nice mix of blue and yellow, whatever, right? But all of a sudden you shake out just a little bit survives. But let's say on just random chance, most of the blue survive. Well, those are the surviving individuals. Well, the new future generations will definitely be more blue because those types survived that event, which then they might lose on a lot of genes that the yellow ones might have had in them. And this is the video. Please watch the genetic drift and founder effect. I don't know what's going on with my computer. There we go. But please watch that. It is absolutely essential. So we get to this idea that population viability analysis, right? There is a minimum viable population. Is the minimum population size required for long-term survival of a species, right? If we have 3 individuals left, it's not going to be a viable long-term survival. We might keep them alive, but they're going to need a lot of human intervention to continue to keep that species alive until so many generations and they start building up mutations of their own. The number of grizzly bears in North America dropped from 100,000 in 1800 to now 1,200. The animal's range is just 1% of what it once was, and the population is fragmented into 6 separate groups. So now where you had 100,000, you're now breaking into 6 groups, and now you only have 1,800, so divide that by 6. So really, you only have about 300 grizzlies in each given area. Well, obviously 300 is not a lot. There's probably not a lot of genetic diversity going on there. Thus, you're going to have grizzly bears that are just not susceptible to fight off infections or diseases like a larger group could. And then biologists need to know how small the bear groups can be and still be viable in order to save the grizzly. So like I said, Elon Musk and his scientists, and I think science in general has dictated that 10,000 humans is our viable analysis. So, roughly what would it be for other mammals? Probably around the same amount. And if we only have 1,200 now, obviously bears are in trouble, right? So it's important to understand these things. Metapopulations are connected populations, right? A metapopulation is a collection of populations that have regular or intermittent gene flow between geographically separate units. We need to provide it something like this bottom right photo, right? If we do have their islands fragmented, like the bears live in different one of these, we have to be able to allow the flow of genetic diversity between them. So we have to let them have passage between each of these given areas. So source habitat. That's the birth rates are higher than death rates. Surplus individuals kind of migrate to new locations. So you have the source here probably, right? And then you have a sink habitat. Birth rates are less than death rates, and the species would disappear if not replenished from the source, right? So you have maybe a lot of individuals growing here. They're dying out here. We can kind of flow them towards there, or they can, migrate to those given areas. And this is kind of just another kind of population, right? But you see the interactions between this island biogeography, right? Of individuals going from one area to given another. And ladies and gentlemen, this is also brings up another problem. Is this why you have more interaction between humans and nature? We actually see in the news a lot, like, somebody was attacked, at least in the United States. I see it all the time where, a lady was jogging and attacked by a cougar, or her dog was, or bear. We can't blame the cougar, right? Or we can't blame the bear, because they are simply trying to probably find a mate or go migrate to a new location. So they often have to go through these human habitats to get to these locations. And then finally, to kind of end this kind of lesson, it depends on the species, right? Each species has a different lifespan. Humans live to be about average roughly 80 years, right? Give or take, depending on the nation. Elephants tend to live about the same long, but we can look at, cats and dogs, for example. They live, maybe 13 years roughly, depending on the breed. But, the maximum lifespan is the longest period of life reached by a given type of organism, right? So, here's some more examples. Bristlecone pine live up to 4,600 years. Humans have been known to live up to 120 years. Microbes may live only for a few hours. So it really varies by species. Differences in relative longevity among species are known as survivorship curves, which is right here. And this really shows you just, how long roughly does the majority of the population live for, right? So we have humans. Well, now because we have healthcare, less violence, we don't have predators, humans are born, right? Here's where they're kind of born, and most of them survive for relatively long. And then all of a sudden they hit about 70 or 80, and then they start going down. That's when most of them then die, right? If you're looking at natural lifespan. Where birds, they have predators to deal with. So a lot of them are getting eaten throughout their lives, and so their curve is more straight to the point, kind of way of putting it, right? So many will die. Then they start losing their odds of surviving decrease as the days go by. Plants, on the other hand, tend to fit this type of curve where plants put out tons and tons and tons of seeds, right? So they all kind of sprout, this growth sprout, but as they start to get bigger, they quickly die off because they're either eaten or they don't have enough sunlight. So when they get to relatively older age, when they become a tree, well, then they tend to survive a lot longer once they hit that certain age time. And finally, ladies and gentlemen, go back to that. How gods die. I want you to please watch that video on just what, explaining once again what happens if you're on a small kind of, carrying capacity and when you overgo what you can do. And I think we already kind of watched that, but you can go ahead and watch that again. And then we will have another lab associated with this, but this is a virtual lab, and we'll talk about this more on your future times. But please just do the actual lecture. Hopefully you watch this to prepare you for the exam and quiz. All right, everybody, that is it. I won't bore you too long. Please have a great day. Enjoy and have fun.